Are you seeing that?
Yes.
Okay, fantastic. Well, thanks so much. And it's really nice to, I really appreciate you inviting me to give a talk here. And this work is just so satisfying and has been such a lovely piece of work to be involved in. So I just love talking about it. So I'm going to talk you through just some of the basic base model and the modeling approach that we published in Nature towards the end of last year. And then towards the end, I'll talk a little bit about further extensions, integrating more types of data into the same kind of modeling technique. But what are we talking about? We're talking about a kind of multi-disease continuous time generative transformer model called Delphi 2M. And this is typically the type of model that's used in large language models like ChatGPT, for example. But we made some key innovations in terms of the architecture of the model. And that's what I'm showing here. So this is the only kind of AI architecture diagram I've got in the whole talk, but it's kind of key. And what you're looking at there is a classical transformer, a decoder only transformer. And you can see some features, some classical things like these causal attention mechanisms and other things, but I've highlighted in red the differences to the standard, or some of the differences to the standard modeling architecture that we've made, which are important. And in particular, this time to event head and this age encoding. And that what this allows you to do is represent information on a continuous time axis within these models. And for health modeling, that's really important. So we're replacing the traditional kind of sequence-based encoding with an age-based encoding. And that allows you to predict not only what is likely to come next in somebody's healthcare journey or their tokens of health, if you want to put it that way, but also when that's most likely to happen. So that's one of the key differences to the standard GPT style models. So we were really fortunate, we have the access, and there's many fantastic research cohorts around the world with large amounts of linked data, healthcare records and genetics, for example. And we have worked for many years with the UK Biobank, which is a fantastic UK-based population cohort with around 500,000 individuals with a very large array of linked information, genetics, healthcare records, and many other phenotypic measurements. And we used that cohort to train our model, splitting the data in this kind of traditional 80% training to 20% validation. But we also held out some information later in time, which allowed us to do some longitudinal validation. And we have long-standing collaborations with colleagues and friends in Denmark, and were lucky enough to get access to a Danish health registry data set made up of about 2 million individuals from the Danish healthcare registries. And we were able to use that as our external validation cohort. And what we're really talking about in the very basic Delphi model is we're only using healthcare information across somebody's life with some basic demographic information such as sex and some limited amount of lifestyle measurements. And that's really all we're talking about in terms of what is fed into the model and trained, and in a kind of next token predictor manner. So we're trying to, rather than predict the next word in a sentence, we're trying to predict the next event and the timing of that event within somebody's health trajectory. Okay. So remarkably, and it's really satisfying to see, actually, with the innovations that we made and some of the training methodologies, we are able to get a really high level of predictive accuracy. Here we're looking at the AUC value, so this is the area under the ROC curve. It's a measure of predictive accuracy for the next token within somebody's trajectory, and you can see that we every dot here is a disease, and it's colored by the ICD-10 chapter. And you can see that across the board, we actually have really rather good performance for a broad set of diagnostic codes. And that's quite remarkable, really, that you have a single model that's able to predict with high accuracy such a large space of disease. The model that I'm showing here includes over 1,200 diseases, all within the same model and mapped to continuous time. And if you look at the distribution across different ICD-10 chapters, you can see. Oh, was that? Did you want me to stop or no?
No, I am not sure who's.
With that, that's okay.
Yeah. Everybody, please unmute yourself. If I can figure out who it is, I can unmute them.
So anyway, so that's fantastic, and you can see that there's a bit of differences between the ICD-10 chapters, but actually on the whole, we're pretty consistent across the board. And if you look at, if you look at the in aggregate measures across all diseases between males and females, there's also no sex bias, which is nice to see. Okay, so so that's kind of remarkable, the overall performance of the model across such a large set of diseases. It was really amazing that we had the access to this external validation data set, which is a totally different healthcare system, information on close to 2 million individuals from the Danish healthcare registry. And we were able to use that as a zero-shot test set. So we were able to evaluate the model trained in the UK Biobank, looking at information from the Danish registries without any retraining or fine-tuning of the model. And that's all summarized here in one slide. But actually across the board, we see a marginal drop in accuracy across most diseases as expected, but actually not a huge drop, which is quite, it's really, really nice to see and quite remarkable. So it shows you that the model was able to learn some general patterns related to health, which can be transferred across at least 2 different healthcare systems in 2 different countries. And if you look at the correlation between individual ICD-10 codes, looking at the AUC values from UK Biobank against Denmark, you can see this remarkable correlation. So essentially, we're able to predict well pretty much the same disease classes across the 2 different healthcare systems, which is really nice. So one feature of the model that I kind of touched on was that we have this internal mapping to continuous time. And that means that it's very natural for the model to look at longer or shorter distances into the future and to represent information based on the time domain. And here we're looking at the delta AUC. So this is the difference between the predictive performance for the next token in somebody's healthcare trajectory against 10 years into the future. And you can see that, as expected, all disease classes decrease in accuracy over time, but actually very variably so. And for some diseases, we remain highly accurate many years into the future, even up to 10 years, which is really remarkable. And if you look at the decrease in performance in aggregate across different time windows, of course, it decreases across time, as you would expect, but actually it does remain relatively stable for many conditions for, up to 10 years or more into the future. So another nice feature of this model, because of the way that the logits are represented within the output of the model, we can represent that information as rates. It's very natural to think about those probabilities as rates. For example, the rate of disease per year. And that's what you're seeing here for 10 selected diseases out of the 1,200. And what you see is every dot here is an individual colored by either male or female across their age range and the predicted rate of disease for those individuals at any point in time, or at a specific point in time. And the lines you're seeing is the actual reported instance, so the actual instance of disease. And there's a few things that you can see. So, for example, on the whole, most of the model's predictions mirror the incidence rates quite well, but there is some spread, and that is a nice feature of the model. Actually, we like to be able to separate individuals at higher or lower risk at any point in their life, and a lot of the kind of patterns that you might see here make sense. So, for example, diseases like Alzheimer's disease or death are increasing much more later on in life, whereas childhood conditions like chickenpox are highly likely earlier on in life and then tail away. And you can see kind of characteristic sex effects like for breast cancer here, and maybe another one to highlight is depression, which is low earlier in life, raising up in risk towards late teenage years, and then remains relatively stable across time. And then finally, for heart conditions, you can see this characteristic increase in probabilities or increase in rates at around the age of 40 or so, with the known sex bias. So that's really nice, and it's a really nice way to be able to represent the information that's predicted within the model. So another cool feature about these types of models is that they are generative, so you can actually simulate data from any starting point up to any point into the future. And so we did that. This little diagram here depicts that kind of idea. So you can have some starting point, some tokens or some information on some people for a particular point in time, and then you can simulate what's likely to come next through the generative process of the model. And then, for example, compare it to what was actually observed. So that's quite a nice test to be able to do. And that's what we did. So we actually simulated data from birth. So only providing the sex token and some randomization of the lifestyle information, and then evaluated the disease rate from the simulated data between the ages of 70 and 75 against what was actually the rates that were actually seen at 70 and 75. And what you can see here is this remarkable correlation in rates across all diseases, which is extremely satisfying to see. And actually that's really quite difficult. So generating fully synthetic sequences that far into the future is a hard task. And what this shows you is that the Delphi 2M model was able to learn some information about rates and timings of disease which mirror real life, of course, but is able to generate fully synthetic data sets from that model. And that has a number of interesting, useful applications. What you can then go on to do is train a new model based on only those simulated data. We did that and compared it to the performance in the original model, and you can see that overall there's a drop in performance, but actually still rather high predictive potential from the model, and that's based on purely synthetic data. So that's kind of really cool. Here's another just representation of what the model actually, how Delphi thinks about the world and thinks about disease. So this is just a 2D representation, a UMAP of the embedding matrix, the internal embedding matrix of the Delphi model, colored by ICD-10. So each point here is a disease. It's colored by the chapter, and the point size is related to the number of entries within the training data set. And you can see a few different features here. So typically diseases of the same chapter tend to be closer to each other in space, which one might expect, but it's quite nice to see that the model is able to do that. And some other kind of key features maybe is this ominous token here, the death token, is kind of clustering closer in space to some acute conditions like heart conditions and respiratory events. And it's worth worth pointing out that actually what happens from Delphi's point of view as somebody ages, they essentially move around this space. So that's just one way of thinking about it. So that's kind of nice. Another way that you can think about, what the model's actually thinking about interactions between different factors, different diseases, is look at this kind of SHAP analysis, which is essentially a sampling technique which allows you to look at the importance or the impact that a token has on the prediction of another token or disease, is of course in this case. And what you can, so it's kind of a two-way thing, predictor versus predicted. And what you can see here is that we have, again, most of the ICD-10 chapters are close or are impactful to each other. So once you have one disease within a chapter, the model thinks it's very likely that you're going to get another one within that chapter for a period of time. By the way, these are time resolved, so this is looking at within the next 5 years. You can look at very specific windows if you so wished. And another feature to highlight is things, acute and serious conditions like cancer, for example, are highly correlated with death, sadly. So that is basically a very quick look at the main results, I would say, from the paper. That paper is very extensive. We did a lot of additional analyses. So definitely do read it if you're interested. I think it's a great presentation of this, the power of these types of models. We have been working for some time on extensions to the model. The basic framework is very flexible, and we've added a lot of more general frameworks to allow you to add additional data types, and are actively working on this. So, for example, addition of prescription records within the same model, further diagnostic tests, biomarkers, genetics, and even lifestyle exposure measures. And so we're working on multiple angles here. And of course, clearly these models are useful for predicting disease, but they have a whole range of other really important and impactful application areas, such as drug discovery, even genetic analyses like GWAS tests, and other things. So I'm just going to really go quite quickly for the rest of the slides, and these slides are a bit old actually, but I just wanted to show you some examples of adding additional information into this type of model. So as I said, Delphi is a very, very flexible framework. We can add pretty much add any data type that you could imagine. But here I'm going to talk about adding genetics and drugs into the model, and you can think of that as G and E. So genetic component and the environmental component, which is drugs basically. So we chose, in this work, we chose to focus on one particular source of genetic variation, which is the HLA complex. And for any of you that have done GWAS or know genetics, this is a really important genetic region. It has very large blocks of LD and typically tend to be inherited together. It's a really complicated region and difficult to call genetically. But whenever you run a GWAS, almost all GWASs, you will find this very strong peak on chromosome 6, which is essentially the HLA region. So essentially these variants associate with many different diseases. So we thought that it was a good one to start with. And as I said, these regions are quite complicated. There's lots of variation and lots of different genes which are inherited typically inherited together for many generations. And it's essentially this protein which is which kind of captures or presents proteins to foreign invaders like bacteria and viruses. And it's very important in immune diseases. But you can represent the alleles across this region in a number of different ways. The most simple is just one hot encoding, which is what I'll show you the results from. So we basically have a lookup table that shows you every possible allele combination, and that is then mapped directly into this transformer or a Delphi style model as an additional token set. So what you're seeing here is so we did that. We added HLA information into the model using very similar framework from before. So the same kind of idea with previous healthcare records. And here you're looking at the delta AUC values again for a model that includes the HLA alleles versus one that does not. And you can see kind of remarkably that across the board, we get generally an improvement in accuracy with looking at AUC values for most diseases, actually. And there are some where we have really quite a big impact in terms of knowing something about this source of genetic variation. The model is able to leverage that information to improve its ability to predict disease into the future. And actually, if you look at which diseases are these the higher impacted diseases, it makes quite a bit of sense. There are many autoimmune and infectious diseases and also cancer, which makes total sense. And actually, one of those is this psoriasis condition. And here I'm showing you a time-resolved AUC plot for males and females separately. And what's quite nice in this case is that we have a much better ability to predict psoriasis early in life, having added the HLA information, and then that gets more marginal later in life. And it's known that the genetics of psoriasis is impactful for early psoriasis compared to late psoriasis, whereas late psoriasis is kind of more impacted by other factors such as previous health conditions and other things. So that's really quite nice to see. So then what about the E component? And again, these are old slides, but I'll just show you just as an example how this looks. So we were able to map all of the prescription records into a particular coding system known as ATC. And we were able to do that for all drugs, but in this case, we're looking at cardiovascular drugs and central nervous system drugs as a kind of case study. So we were able to train another model that now includes also health records, but also drug prescriptions across time for cardiovascular and nervous system drugs. This is a view on just the internal representation of that model with drugs colored in purple and cyan. And actually, largely, you can't really interpret this space, but the drugs are in there. But one way that you can have a look at the impact of inclusion of drugs within the model, again, we could look at this SHAP analyses. And here we're looking at the same kind of heat map, but again, with drugs included alongside all the ICD-10 chapters. And what you can see on the whole is that actually the prescription of one drug within this drug class is impacting the model's ability to predict that you will be prescribed another drug within the same class, which maybe not too unsurprising, but actually if you look carefully at the structure underneath this, for central nervous system drugs, you have either the prescription of a drug impacts the diagnosis of mental health conditions and central nervous system disease, and for cardiac drugs, cardiac disease, which again you would expect. But actually there is an interesting aspect here, which is we're training the model to predict disease or predict drugs, but actually often drugs are either used as a treatment for a disease or as a preventative. And so the timing of those two things can be quite interlinked, or put it another way, can be before or after each other, which kind of decreases, if you're looking at the ability to predict, it's not so black and white. So one way to look at this is, for example, if we now look at drug and disease pairs, and the variance. So the mean predicted SHAP value for drug disease pairs versus the variance in that SHAP value. And essentially what you would like to be able to see here for a drug disease pair, which is impacting the or you would like to see those robust predictions over here, essentially. And you can see that there is some, so essentially what you're saying is do cardiovascular drugs, or which cardiovascular drug increases the predictability of a cardiovascular disease, those shown in red, or versus the other conditions. And here I've just shown mental health disorders and nervous system. And of course, what you would like to see for an improved performance metric is that a large majority of the cardiovascular conditions are over here on this side. But actually, you could also see that perhaps a drug was protective, and that would be on this side. Okay, so the prescription of a particular drug decreases the likelihood of you having a condition overall, and that would be over here. Okay, and although it's a bit difficult to see in this plot, there is a significant shift between cardiovascular disease and the other conditions. Now, if you look at central nervous system drugs, this effect is even more clear. So if you have been prescribed a particular central nervous system drug, then your likelihood of being diagnosed with a particular central nervous system or mental health condition much higher from the model's point of view. So that's kind of neat, and I really just wanted to show that as one way of picking apart this information. It is actually rather complicated, and particularly to do with the timing of prescription versus diagnosis and a whole bunch of biases that go into that. So it is actually quite challenging to pick apart. But you can, for example, look at what these drug pairs are. This is just showing that this is a particular drug. It's ketamine. It is also the frontline treatment for schizophrenia. So that's quite nice that we see this is one of the outliers. And here is another drug which is associating to many different diseases. And this, I believe, is actually like a broad spectrum pain pain-related drug, so it's essentially being prescribed for many different disorders. Okay, and of course, what about other diseases? So, as I said, it's quite a complicated space to explore. But what one might want to look for are adverse drug reactions. So, a drug that is being prescribed that is then impacting the prediction of a disease which is outside of the scope of the drug, for example. That's an interesting area to explore. And also these types of protective drugs. So once prescribed with a drug, the model thinks it's very unlikely that you will get a particular condition. So those are all areas that we certainly are looking into, and there's lots of potential to look at that type of thing. So yeah, so in summary, we developed a model. It's remarkably performant. We're super happy with it, and it allows you to essentially align whole populations across space and time within a single modeling framework that is optimized for disease prediction across a large spectrum of disease. And I like to think of it as a kind of general framework where you can align lots of individuals, but also lots of different types of data sets, all mapped to disease prediction in whole populations across time. And yeah, these techniques are have a huge amount of potential, both for scientific research, but also for other direct clinical applications and more. So I just acknowledge the key people within this work. My colleagues at the EBI and at DKFZ, and in particular Moritz Gerstan and Artam. And then we were very lucky to have long-standing collaborations with people from Copenhagen, Soren Brunak and others, which allowed us to have access to the Danish healthcare registries and to perform some analyses with them. Thanks very much, and I'm very happy to take some questions.
Thank you, Tom. This was awesome. And what is very remarkable is that people build these predictive models for like one disease at a time. And what you have found is that your model often compares with this disease, the AUCs compare with the disease specific models, right? So it's really amazing. So I'll open it for questions. I also have a couple, but I would invite others to ask theirs first.
I mean, also, I mean, thanks for highlighting that. I didn't have those slides in the deck, but we actually, as a consequence of the review process going through Nature, we had to compare the model to all types of single disease risk models and other models that you could possibly imagine. And actually, it was really satisfying to see that actually on the whole, we were either as good, if not better, than single disease risk models, a lot of which are either clinically implemented or have been kind of handcrafted over time with real expert knowledge. So that's really kind of neat.
It's remarkable. Bryce?
Yeah, thank you. And thank you again. I'm curious, with your model accounting for the age across the, an individual case, have you, or do you see any value in or ability to account for age of kind of source data to be able to track how correlations change through, through our real time advancements in healthcare, environmental changes, lifestyle changes across populations?
Absolutely. I think that's a really insightful question. So we can easily change this from age to year and have models that are really tracking the evolution of healthcare availability and risk of disease across many years into the future, or many years into the past even. But I guess the challenge there is, as one might expect, data availability, right? So we only have electronic health records for a certain amount of time back in time. Going forwards, one would think that you would absolutely want to have these types of models which are tracking patterns either in one population or ultimately in many populations, and being able to look at those trends across the world would be super cool. But it certainly has that potential.
Thanks.
Thank you for the question, Bryce. I was kind of thinking the same thing, because when you're making a 75-year-old prediction, that prediction is made on the data that's available right now, right? But who knows what else is going to change in the next 75 years by the time this person is 75 years old. So yeah, thank you.
Yeah, absolutely.
Other questions?
I saw one in the chat, which is, I'm curious if you added a contrastive loss function for the drug disease pairs to encourage separation in the embedding space. It's noisy where I am, so I can't unmute. That's a really good question. Actually, no. So, but it's something that's gone through my mind. So the slides that I was showing you were quite basic, really. It was, as I said, they're quite old. But you could definitely improve how to model drugs within the framework. And one other way that I've been thinking about it is to have a kind of encoder-decoder architecture. So basically, you want to learn the relationship between drugs and disease, where encoders and decoders are, that type of architecture is quite often used as in language translation. So when you have to map one kind of language to another, and you could think of drugs being one language and disease being another. So I think that's also an interesting area to explore.
Mike? Doesn't look like Mike has a question. So, yeah, so I have a question, Tom. So in the one of the very first slides where you talked about the differences between the transformer architecture, like default, and what you are doing, there was something about the causal nature or causal structure, causality of what causes what. Can you elaborate on that, please?
Yeah, I can a bit. I mean, it's it's quite interesting. So in the AI field, causal means something very different to what it means in biology. So causal attention block is basically saying we can't look into the future, right? So you're not allowed to look into the future. It means it's causal. That's and so there's always a kind of confusion there, and it's worth that causality question is really interesting. You know, these models, or the model that I presented, you have to be quite careful about interpreting interactions between factors as being a causal link, because the model's just trying to learn and turn levers in a way where it's able to leverage all information to improve its prediction. So it's not naturally a causal model, but what you can do is, and this is something that we have done in a variety of different ways, is try to force the model, or to encourage the model to actually look for real causal links between factors. And that's a really interesting space. I would call that causal attention.
Got it. So what you are doing is that just not letting future affect the past, because in language, like the second half of the sentence can be used to predict. Like when you do the masking, it's not necessarily just the words that come before that are predicting the next word, but in the attention mechanism, it's all to all, right? So it's correlated, everything is correlated. But here you are kind of masking it somehow so that there is.
Yeah, we use the typical way that these models tend to be trained, which is causal attention, which means that the events into the future cannot be seen by the model. But I think, you know, if you have an encoder decoder architecture, that is somewhat merged a bit more. But yeah, so it's the classic kind of next token predictor causal attention.
Got it. And how would you figure out the causality, as in like just correlation versus causality?
Yeah, well, I mean, I think that's an area for for innovation, an area that is ripe for innovation, and there are a number of ways that one could think about doing that, leveraging things like genetics and other factors to try to encourage the model to learn those known causal relationships, for example.
Thank you. Any
Pleasure.
more questions? If not, I would love to thank Tom again. Thank you so much for making the time to come and talk to us. This was really cool. They just, it's such important work and far reaching, so many applications. So actually that makes me think of one more question. So what is happening with this? Like, so are people kind of taking this and trying to convert it into some sort of, product or something that can be rolled out to clinics? What further steps are needed before it's applicable in clinics?
Yeah, I think that's a great question. It's a question that we've been asked so many times over. But I think, in terms of rolling this type of model into clinical practice, I think there's a lot of people who are kind of make grand claims about that. But actually, there's quite a lot of legwork that needs to go on. For example, regulation, how best to integrate these models in a way that they provide maximum benefit while avoiding any potential real or perceived harm. And so I think that's definitely, there's a lot of activity going on around policy and regulation for these types of models. And, on the whole, understandably, I think organizations and healthcare systems are somewhat cautious about implementing these models. So I would say that they have huge potential to benefit health and benefit. So there's one thing about predicting health. You could imagine these types of models being used as a kind of clinical assistant, if you like, or an early warning system within clinical care. That's a kind of obvious way of thinking about it, and should help clinicians and therefore help patients in the end to get earlier treatments and hopefully better outcomes. But there's another part that's potentially a bit less obvious, but also makes a lot of sense, which is to do with healthcare planning and knowing across, you know, a population or across a subset of population what disease is likely to happen at any point into the future, and therefore be better prepared and better able to plan those important health resourcing activities. And I think that has a real potential to improve healthcare resourcing and planning, and therefore delivery. So, but I think when, you know, it's probably 5 to 10 years out, really, or who knows, maybe you'll see them in certain clinics, but I think it, you know, before they're rolled out worldwide, and you see them very, very commonly, it's a number of years off, I would say.
Yeah, thank you. And that's a great point, that on a population level, there are probably more and safer use cases to kind of predict trends on a population level. But predicting this on an individual patient level, like one can think of potential downsides to it also, right? Like if I know that, okay, I'm X likely to get that disease, like for 1,000 diseases, that can easily cause me to worry, and like that, like the if there are false positives, that can be really, really bad. And also if this information like goes to the insurance companies, like they can, like that can change the landscape there as well. So
Yeah.
and that can harm patients potentially.
Yeah, absolutely. So that's why it's important that these regulations are thought through carefully, and that there's policies that are put in place, and people need to understand. And I think people generally do, you know, that these types of models are predictive. They're not certainties. You know, I mean, it's a bit like predicting the weather, isn't it? That people will know that you can predict that it might be sunny with some accuracy tomorrow, but to predict that, you know, at a particular day 2 years into the future is obviously very much more challenging, right? So.
Right, and it doesn't mean you should sell your umbrella, probably.
Yes, well, exactly. Yeah, exactly.
Okay, any final comments or questions? Okay, thank you, Tom. This was great. Thank you very much.
No, real pleasure. Lovely to meet you all, and yeah, have a good day.
Good night. Thanks.
Thank
All right.
you.
Take care. Bye.